My name is Christina Manuel, and my topic for the mini lecture is leptin. This lecture will discuss the function of leptin regarding physiological functions as well as endocrine functions. I will also be talking about the effects of leptin on different species that we've discussed throughout the semester, including beef cattle, dairy cattle, swine, and sheep. What is leptin? Leptin is a 16 kilodalton polypeptide protein containing 167 amino acids. It is a pro-inflammatory adipokine, and adipokines are cytokines produced by the adipocyte. And cytokines are small proteins involved in cell signaling. Leptin is a hormone made by adipose cells, and it's synthesized and secreted by white adipocytes in proportion to the amount of lipids stored. It causes the release or the inhibition of factors that help regulate energy balance by inhibiting hunger or increase energy expenditure in physical activity. And the primary function of leptin is the regulation of energy. Is known as the satiety hormone, and it's named from the Greek lepto meaning thin. It is the first fat cell derived hormone or adipokine, and it was discovered in 1950. So it's made by adipose cells and inhibits hunger, and is opposed by ghrelin, which is known as the hunger hormone. Leptin acts on the receptors of the hypothalamus to regulate appetite and achieve energy homeostasis. And again, its primary function is the regulation of fat stores. However, leptin has other roles in physiological processes because it is in multiple sites of synthesis other than the fat cells, and there's multiple cell types besides the hypothalamic cells that have leptin receptors. So leptin was identified in 1950 using the obese mouse model. So mice that were homozygous for the obese mutation became obese, and in 1960 a second mutation with the diabetes gene was discovered, and those homozygous mice were also obese. However, in 1990 the gene mapping of the obese gene discovered that that gene encoded a hormone that circulated in the blood, and that hormone suppressed food intake and body weight in the obese and wild type mice only, not in the mice with the diabetes gene. So in 1994 leptin was identified as this hormone, and in 1995 there was evidence that the mutations in the obese gene in the mouse did not occur in humans. In 1995, further studies confirmed the diabetes gene encodes the leptin receptor. So where is leptin found? It is primarily produced in the adipocytes of white adipose tissue, but it's also produced in several other places in the body, including brown adipose tissue, the placenta, ovaries, skeletal muscle, the stomach, mammary epithelial cells, bone marrow, gastric chief cells, and the PD-1 cells, which line the fundus of the stomach in humans. So this slide is showing a few examples of the actions of leptin throughout the body. So leptin is found more in the subcutaneous fat than visceral fat, and it's stimulated by insulin, glucocorticoids, tumor necrosis factor, estrogens, the CCAAT enhancer binding protein alpha, and leptin is inhibited by the list in the red with androgens, FFA, growth hormone, and peroxisome proliferator activity receptor gamma agonist. And the primary function of leptin is to increase satiety and energy expenditure through the action on the receptors on the hypothalamus. So in the muscle, leptin action increases glucose uptake and glucose metabolism. In the liver, leptin increases glucose production, and in the pancreas, leptin increases insulin. So I'm first going to talk about the physiological function of leptin, and this includes maintaining energy homeostasis, and that is the energy intake and expenditure. And it also includes the metabolic signal of sufficient energy rather than excess energy, which is the primary role of leptin. How does leptin work? Leptin is the afferent signal in a negative feedback loop, and it maintains the homeostatic control of an adipose mass. Leptin travels in the blood and signals the brain to regulate food intake. When fat mass decreases, plasma leptin concentrations in the blood also decrease. This stimulates appetite and suppresses expenditure of energy until the fat mass is restored. However, when the fat mass increases, leptin levels also increase, and this suppresses appetite until weight is lost. Leptin acts on the receptors in the hypothalamus and in the brain to regulate energy balance and other systems. Also, the amount of leptin expressed by adipocytes correlates with the lipid content of the cells. So once leptin is synthesized, it's secreted through a constitutive pathway and not stored in the cell. And mechanisms for regulating leptin expression in adipocytes is still unknown. However, it's likely a number of hormones modulate the obese gene expression, including glucocorticoids and insulin. Leptin and the regulation of body weight in mammals was looked at by Friedman et al. In the Nature journal. The cloning of the obese gene in leptin indicated that body fat content may be under homeostatic control, and the level of leptin is positively correlated with differences in body fat. Increased leptin results in negative energy balance, so an energy expenditure is greater than food intake. Decreased leptin levels lead to a positive energy balance, where food intake is greater than energy expenditure. Also, leptin acts centrally to decrease food intake and modulate glucose and fat metabolism. So there are three general ways in which alterations of leptin in the regulatory feedback loop can lead to obesity, and they're seen in the figure on the right. So the first one is where the letter A indicates absent leptin. So if there's a failure to produce leptin, like in the homozygous obese gene mice, it would result in obesity. Also, with a regulatory defect, so that would be a low leptin secretion for a given fat mass, and the fat mass would expand until normal leptin levels are reached, and this also results in obesity. And the third way is leptin resistance, where obesity could result from relative or absolute insensitivity to leptin at the site of action. So this resistance would be associated with increased circulating leptin, and that's analogous to the increased insulin level seen with insulin-resistant diabetes. So this slide was taken from an article in the Nature journal, also by Friedman et al., and it looked at leptin and the regulation of adipose tissue mass. So it's showing that at an individual's stable weight, which 15 to 20% of the body is fat, the amount of leptin circulating in the blood elicits a state where the food intake equals energy expenditure. And these effects maintain consistency of fat cell mass within a narrow range. However, the intrinsic sensitivity to leptin is reduced among the obese, where the body fat content for their weight is 30 to 35%. So the obese individuals will have higher leptin levels, and they enter a state of negative energy balance when the weight is reduced and leptin levels decrease. Okay, so next I'm going to talk about the endocrine function of leptin, and this includes neuroendocrine function as well as leptin has a role in immune function, hematopoiesis, angiogenesis, and bone development. Describes the endocrine function of leptin. So you have an endocrine adipocyte with several proteins and enzymes that are involved in steroid metabolism, and they are seen on this picture on the left, including angiotensin, leptin, adiponectin, and several others. And the picture on the right shows the receptors found on the adipocyte for these different proteins and enzymes. So combining the specific receptors and the production of these endocrine enzymes and proteins, the adipocyte will perform specific functions in metabolism. Also, the different adipose deposits may function differently depending on what's produced and affecting various tissues. So for example, leptin is produced more in the subcutaneous fat than visceral fat. And also the location of the adipose deposition, such as the visceral adipose tissue, will secrete endocrine substances into the portal system with direct access to the liver, whereas the subcutaneous adipose will secrete substances directly into the systemic circulation. This slide is showing leptin's role in angiogenesis, which is an example of an endocrine function of leptin. So leptin directly induces angiogenesis, which is the development of new blood vessels, by activating its receptor, which is the OB/RB, which is expressed on the endothelial cells. So leptin is also able to induce vascular fenestrations and permeability. So the next section of the lecture, I wanted to discuss specific effects leptin has on different species, including the beef cattle, dairy cattle, swine, and sheep. A study by Lagonagro in 2003 discovered a new mutation in the coding region of the bovine leptin gene associated with feed intake. And the researchers took a cattle population and screened them for polymorphisms in the leptin gene, and 5 single nucleotide polymorphisms were found in the regions containing the coding sequences. So researchers evaluated the association of these polymorphisms with feed intake and fat-related traits. And the results suggest an association between a polymorphism in exon 2 and feed intake. And so the cattle with the genotype AT at the position in exon 2 had 19% greater mean feed intake than individuals with the genotype AA. And results from this study also provide evidence for a link between leptin haplotypes and fat-related traits. Leptin in dairy cattle was examined in this particular study by Liefers et al. In 2005, and researchers examined the genetics and physiology of leptin in periparchment dairy cows. So with dairy cattle, the increase in milk yield has a greater negative energy balance during early lactation and a decrease in fertility. Researchers determined the function of leptin during this periparchment period had polymorphisms in the bovine leptin gene, the leptin receptor gene, and fertility and production traits. Results of the Liefers study determined leptin concentrations were high during late pregnancy and declined to a nadir at parturition. The two extreme genotype combinations could be used to investigate the function of leptin concentrations in the pregnant cows, and the two genotype combinations of the three single nucleotide polymorphisms were defined as having good milk yield with a good energy balance and fertility. Genetic analysis of the leptin gene also indicated a combination of three polymorphisms located at 135 base pair region of the leptin promoter, which explained most of the variance in the prepartum leptin concentrations. There was also a polymorphism on the intron 2 of the leptin gene, which explained a significant part of the variation in the milk yield. So calculations will determine if one of these genotype combinations would be a possible option to use in selection for dairy cattle. A study by Barb et al. In 2001 discussed the biology of leptin in the pig. The porcine leptin receptor has been cloned and is in the class 1 cytokine family of receptors. In pigs, the leptin receptor is in the brain and the pituitary. So, in vitro studies showed the leptin gene is expressed by porcine preadipocytes. The gene expression of leptin is highly dependent on dexamethasone-induced preadipocyte differentiation. Hormonally driven preadipocyte recruitment and subsequent fat cell size may regulate the leptin gene expression in the pig. Further results of this study determined adipose tissue leptin gene expression in the fetus may be increased by adipogenic hormones indirectly. Leptin administration to the pig suppressed feed intake and stimulated growth hormone secretion. Also, the serum leptin concentrations increased with age. And results showed the estradiol-induced leptin, mRNA expression, and fat in the pre-pubertal gilts was dependent on age and weight at the time of expected puberty in the intact contemporaries. And this was also associated with greater LH secretion. Research also showed leptin acted directly on the pituitary cells to enhance LH and GH secretion, and also the brain tissue to stimulate gonadotropin-releasing hormone secretion. With these results, developing proper nutritional plans or gene therapy can manipulate the leptin secretion and help determine practical methods of controlling appetite, growth and reproduction within the farm animals, and it can increase the efficiency of lean meat production. Finally, I wanted to discuss leptin in sheep, and the first study is by Blasiak et al. In 2015, and they looked at the role of hormones and growth factors in initiating and maintaining the lactation of seasonal animals. So lactation requires numerous hormones for processes such as lactogenesis and galactopoiesis. So specifically, researchers studied the effect of leptin in mammogenesis, lactogenesis, and galactopoiesis. The leptin mRNA in the mammary gland cells of many species, including humans, suggest that the mammary gland may be the structure producing leptin. Well, further research of the endocrine mechanisms facilitating the maintenance of lactation during the autumn and winter may contribute to the improved profitability of sheep's milk. By Long et al. In 2015, researchers evaluated differences in adipose tissue metabolism and function in adult male offspring from obese and control-fed mothers that were subjected to an ad libitum feeding study. The obese ewes were fed 150% of feed provided for the controls from 60 days before mating. The F1 male offspring were fed only to maintenance requirements after weaning until adulthood. And then they were fed ad libitum for 12 weeks with monitoring the feed intake. Following the feeding study, the offspring were given an intravenous glucose tolerance test they were necropsied, and the adipose tissue was analyzed. During the feeding trial, the F1 obese males consumed more feed, gained more weight, and became heavier compared to the F1 control males. The glucose tolerance test in this study determined obese F1 offspring were hyperglycemic and hypoinsulinic compared to the F1 control offspring. The necropsies showed perennial and omental adipose depots weights were 47% and 58% greater respectively, and the subcutaneous fat thickness was 41% greater in the F1 obese offspring versus the F1 control males. The adipocyte diameters were greater in the perennial and omental and subcutaneous adipose depots in F1 obese males. Also, expression of fatty acid transporting, binding, and syntheses of mRNA and protein was increased compared to F1 control offspring, and maternal obesity was increased appetite and adiposity associated with increased adipocyte diameters and the increased fatty acid synthesis in the overnourished adult male offspring. And this is just one of the figures from this article showing that the F1 obese males compared to the F1 control males, that there was a plasma glucose concentration increased every 2 weeks from the samples from week 4 up until they were necropsied. The insulin in the samples every 2 weeks from week 4 up until necropsy decreased, and the leptin concentrations were increased from the beginning throughout the entire challenge in the obese males compared to the control males. The conclusions of this study determined the obese offspring had an elevated plasma leptin and showed a greater feed intake. Also, the postnatal lambs from the obese ewes did not show the plasma leptin surge that the lambs from the normal weight ewes exhibited. The adult offspring from the obese mothers had an increased appetite and gained more weight during an ad libitum feeding study, and the increased and decreased neonatal leptin concentrations led to an increased feed intake and an increased body weight gain. So overall, they determined leptin has direct effects on glucose metabolism, and it may have a role in insulin and glucose dysregulation that was seen in this study. So in summary, leptin is a protein hormone that has important effects on regulating body weight, metabolism, and reproductive function. It's expressed mostly by adipocytes. The physiological function of leptin includes the effects of body weight throughout hypothalamic centers that control hunger, temperature, and energy expenditure. And endocrine functions of leptin include there's low leptin in animals with a lower body fat. This is a list of the references and studies used in this mini lecture.